Okay, good afternoon, everyone. So this is another talk in the Advanced Plant Growth Centre seminar series. One that has been arranged kind of last minute, and Mark has agreed to step in here. If you're at JHI, you probably know Mark. If you don't, Mark's been working on potato for a fairly long time. So he completed his PhD at University of Birmingham, and then did postdoc studies at Royal Holloway, University of London, and then Berkeley, California. And then he joined what was the Scottish Crop Research Institute in 1989, not long after I joined, to be honest. So we're both the old men of the Hutton Institute, I think, at the moment. Mark has pursued a fairly single-minded approach to potato over that long time. Well, other crops as well, but potato has been his main love or bane of his life, depending on what kind of day you get him. But the work's increasingly becoming relevant, particularly under current geopolitical scenarios where food security and the ability to feed the world as climate change comes in and population increases is becoming increasingly important. So Mark's going to talk today about future-proofing potato and the mechanisms and markers for global warming tolerant idiotypes. So without further ado, I'll hand over to Mark and let him wow us with the science in potato. Mark, over to you.
Thank you very much for that invitation, Derek, and for the invite to talk here today. I'll just share my screen, hopefully. Is that showing?
Yeah, that's good.
Okay, so yeah, as Derek said, I've been at this site in Dundee for many years, an aerial view of the James Hutton Institute at Dundee, which is rapidly becoming out of date with the new buildings, including the APGC. So why potato? Well, it's a very important crop, arguably the third most important source of calories to humans, and consumed regularly by over 1 billion people. And unlike other sources of carbohydrate and calories, it has other nutritional value. It has nutritionally significant levels of vitamin C, more potassium per gram than banana, and a range of other vitamins and minerals, as well as fiber. Recent studies from the International Potato Centre, just published last year, actually were very interesting, showed that the iron in potato is extremely bioavailable because of the lack of phytate potato and because of the high vitamin C content. So potato-rich diet can actually protect against anemia. So at the James Hutton Institute, we curate a large collection of potato germplasm with about 1,500 accessions of about 80 wild and cultivated potato species. And this is just a small fraction of the available germplasm in the world, but quite significant. And you can see within this, there's a whole range of colours, sizes and shapes, but also tolerance to biotic and abiotic threats, because these potatoes originate from different agroecological zones. So unlocking this potential is only just becoming a reality, because introducing genetic gain in potato is very difficult because of the biology of potato. Most Solanum tuberosum accessions are tetraploids, outbreeding and heterozygous, which means building genetic gain is very difficult and a slow process. But there are new approaches that are rapidly coming online to overcome this. Things like gene editing. Hybrid diploid breeding is a very interesting approach, which means that you can essentially breed potatoes like cereals and rapidly stack genetic gain. And things like genomic selection are going to accelerate improvements in potato cultivars. So there's every possibility that we'll be able to benefit from this treasure trove of genes and alleles that we have in our germplasm collections. But despite the nutritional benefits of potato, most potato varieties are susceptible to environmental stresses. A lot of potato varieties are not very drought tolerant. Salinity is a big problem in quite a few regions. Frost damage prevents early planting and thus shortens the season in many regions for many varieties. An elevated temperature is probably one of the most important limitations of yield in potato. So even as the temperature gets above 20 degrees, the average daily maximum temperature, we can start to see effects on the potato yield and quality. We get heat sprouting, we get chain tubers, we get mist-shaped tubers. All this impacts on usable yield. And so a few years ago now, we wanted to develop approaches to tackle the problem. And we developed consortia to do this, in which we basically threw the whole of our genetics genomics toolkit at the problem. So we wanted to use genetic approaches, QTL mapping. We wanted to look at metabolites and transcripts associated with elevated temperature, and also combine this with physiological phenotyping. All this leading to candidate genes and pathways and a greater understanding of the processes that go on when plants are exposed to elevated temperature, and what makes a resistant and a tolerant plant, and then lead this through to developing markers for breeding. That was the vision, and of course we're still on that journey to some extent. And I want to talk about some of the progress we have made. But one of the things we wanted to do was a genetic screen for heat stress tolerance. But this is quite difficult because we have to do this on a population of plants, different genotypes. And to get sufficient genetic traction, we need to look at 150, 200 genotypes and grow them under controlled conditions. So for a large plant like a potato, this is difficult and expensive, especially with the facilities we had available. And so we decided to use a model system developed by Elmer Ewing in Cornell in the 1980s. So these are nodal cuttings from a potato plant. And if you bury the apical bud in moist compost, you get a small tuber forming in 2 to 3 weeks. So this is a time and space saving system. And it simplifies the tuberization process. And we can see tolerant and sensitive genotypes giving different responses. So we thought this would be a good way of screening for heat tolerance. Of course, we needed a genetic population to do this. And I want to introduce the 06H1 diploid population made by colleagues at the James Hutton Institute in work led by John Bradshaw and Glenn Bryan. And this has been a very great tool for us over the years, and they've helped us a lot with the genetic analysis. And this population, which basically has both Solanum tuberosum and a relative of tuberosum called Thoreca in its pedigree, segregates for many quality and developmental traits. So it's been used in screens for a lot of disease resistances, as well as in collaboration with Glenn and his group for other traits, as I'll explain. So the heat stress screen, we thought it was going to be quick and easy. In fact, it took us over a year working in the dark pretty much. We didn't know if we were going to get any genetic traction on this trait. And so this is a testament to my group, who put in a lot of work to get this done, over 10,000 cuttings. But we did eventually get sufficient reproducible data to do a QTL analysis. And this is one of the 3 main QTL we identified. It's on chromosome 4. And if we look at the genome browser, can identify a candidate gene. In this case, there was an obvious candidate gene. It was a heat shock cognate 70 in this region that we thought would be a good candidate gene. Fine mapping in potatoes is rather difficult, and so we had to use a candidate gene approach. So we looked at the alleles of this gene in the parents of the population, and we found that each parent had 2 alleles, and both the alleles were different in each parent. So we had 4 alleles within this population in total. And so we could look at the genotypic means for each allele. We found that anything with an A2 allele yielded the best, the highest fresh weight. And we wanted to know the reason for this. So we looked at the expression level of the heat shock 70 on exposure to elevated temperature in genotypes carrying the A2 allele or ones lacking the A2 allele. We saw that the A2 allele was turned on very rapidly and very strongly when the plants are exposed to elevated temperature, whereas those lacking the A2 allele were not. In fact, we made transgenic lines using the A2 allele promoter, and we showed the same effect. We saw rapid turning on of the gene in the transgenic lines compared with the wild type. And we also developed an assay to look at membrane damage so we could quantify the heat stress in leaves. So this is an electrolyte leakage assay, which tells us how damaged the membranes are, basically, in a very simple and quick way. And we can see that there's a lot more membrane damage in the wild type plants when exposed to heat than in the transgenics. And over the course of the season, we believe that this protection of the heat shock 70 allele is translated to increased tuber yield as the plant's protected in these cycles of heat and cooler temperatures. And in fact, we see in this glasshouse experiment, we see quite a considerable yield increase in the transgenic lines. Because all this work was carried out and using nodal cutting assays in a glasshouse in Scotland, where we don't really have much temperature stress in the field, although even in other parts of the UK, this can be a problem in warm seasons. But we wanted to test whether these alleles would offer protection in warm, dry climates. And so we conducted field trials with collaborators in Kenya and Malawi, and we grew about 60 genotypes which had the various allelic combinations possible. In fact, we always saw that the A2, A3 allelic combination gave the best yield in trials in Malawi and Kenya over various seasons. And in fact, when we look back at the data that have been collected in the UK, we also saw that the A2, A3 combination gave the best yield. So we think that this heat shock 70 gene is actually a yield gene. And as well as being heat tolerant, some of the O6H1 genotypes that we tested have good virus resistance, which is essential for growing in parts of Africa, and also achieve maximum yield after 70 days. So they grew quickly. So this is also a benefit in these environments. And in fact, we have 5 of these O6H1 clones on the national list in Malawi, and national performance trials are in progress in Kenya. So I think our work translates through to developing cultivars that might be of use in these environments. We wanted to see if this heat shock 70 effect could be seen in a wider range of germplasm. So we looked at some of our accessions from the Commonwealth potato collection. And here we have 26 of these wild relatives. We carried out our electrolyte leakage assay at 20 degrees and 40 degrees, and you can see that there's a wide range of responses in terms of membrane damage at the elevated temperature. Some are quite tolerant, whereas others are quite heavily damaged by this treatment. But if you look at the expression of the heat shock 70 gene and plot that against the damage, we see this inverse, significant inverse correlation. So the higher the expression of the heat shock 70 gene, the lower the damage. And in fact, going back to the heat shock 70 being a general yield gene, we also looked in a GWAS panel of tetraploids. So this is work from Sanjeev Sharma and Glenn Bryan. They've developed a very nice GWAS panel, and when they look at their yield data, they can see a yield QTL in the position where the heat shock 70 maps, again implicating it as a yield gene. And how does this heat shock 70 allele protect? So we looked at the fluorescent, the chlorophyll fluorescence data, which is an indication of the quantum yield of photosystem 2 in plants that are expressed to combined heat and drought. So this is time course of drought and then drought recovery. What we see is in the genotypes carrying the A2, A3 allele, we see much faster recovery following drought stress. So we think that the heat shock 70 is a chaperone, and we think it protects these delicate protein complexes from the effects of stress. But why should the particular A2 allele be beneficial? Well, the actual coding sequence of the gene, the amino acid sequences, are very similar of all the 4 alleles. But when we look at the promoter sequence, which is responsible for regulating its expression, we do see this run of TA repeats, specifically in the A2 allele, that's missing in the other alleles. And these AT repeats have been known to enhance gene expression. And so we did an experiment using a test plant, Nicotiana benthamiana, transiently expressed a gene construct containing the heat shock 70 A2 promoter, and various deletions that we made of the TA, going from the full 10 repeats down to 4 repeats, which we see in other alleles. So we inoculate these constructs into Nicotiana benthamiana, leave them for a day or two to be expressed, and then expose the plant to heat stress. What we can see is that the more TA repeats, so here we have 10, 8, 6, and 4, the more repeats, the higher the expression level, and generally the lower level of membrane damage. So again, we think that the heat shock 70 effect is a gene expression effect. And do we see this in the wild relatives? And this is work that Ray Campbell has done. He sequenced the promoters of all the wild relatives, and we didn't really see a strict correlation with the number of TA repeats, but there are other promoter elements that we think might be correlated with expression level and hence tolerance to elevated temperature. So to summarize what we did on the heat shock 70 side of things, we believe it's a candidate gene affecting yield. We've shown that the A2, A3 combination gives the highest yield in field trials in warm environments, but also in environments in the UK. And we believe that this protection against temperature and drought translates to improved tuber yield over the course of a growing season. We also see the heat shock 70 alleles that are expressed in response to elevated temperature protect against heat-induced damage in wild species. And I should say that we also have in the same genetic screen where we found this QTL, we have other QTLs, and we're trying to pin down candidate genes using exome capture and bulk segregant analysis at the moment. So moving on to another aspect, I mean, heat stress comes in many different ways. You know, you have some constant high level temperatures and then short spells of high temperature. And we noticed during the course of our work that there was a very strong acquired thermotolerance response in potato. So if we took a potato plant growing at 18 degrees and transferred it straight to 40 degrees, which is pretty high temperature for potato, it died fairly quickly, wilting and cell damage. However, if we allowed the plant to acclimate at 25 degrees and then transferred it, the plant was much happier and much less damaged. In fact, we saw this in most of the tetraploids we looked at, and we could quantify this with our electrolyte leakage assay, showing there was much less damage in the plants that have this intermediate 25 degrees C acclimation period. So we wanted to know how much time 25 degrees C was necessary for thermotolerance. And we can see that between 6 and 12 hours, the plants acquire this tolerance. And also, once they'd acquired the tolerance, do they maintain it, or does it wear off after a while? And we see that between 12 and 24 hours after acquiring thermotolerance, you put the plant back at 18 degrees, it then loses it when you expose it to high temperature again. So other things about the acquired thermotolerance, we wanted to see if you needed light for the response, and you certainly do, because if you try and do the acclimation in the dark, it's as if there was no acclimation at all. And also looking at wild species that we know are thermotolerant compared with sensitive wild species, we see that the tolerant wild species don't need acclimation, whereas the sensitive types do. And also in the O6H1 population, there is some considerable variation for this acquired thermotolerance response. However, it's a fairly laborious and low throughput method of assaying this acquired thermotolerance, and so we've not really been able to do a genetic screen for it. But in view of how powerful a protection mechanism it is, I think this is something that we should look at. And I should say we did spend some considerable time looking at transcriptomics and metabolomics of what was happening during this 25 degrees C acclimation period, but we've so far been able to identify any mechanisms for what's happening. I think it's still an area that needs to be investigated further. So acquired thermotolerance is a powerful adaptive response, light dependent, reversible. There's considerable variation in different genotypes, and as I said, we don't understand the mechanism yet. I'm going to change tack a little now, and I'm going to talk about the potato tuber life cycle. And this really is the site of action of many environmental stresses, and how the potato tuber life cycle is regulated is very interesting. There's various tiers of regulation of this cycle, because the plant produces a tuber as a sort of last resort when it no longer thinks it can reproduce sexually, uses these vegetative organs to sort of overwinter and then grow again the following season. And there's a lot of interesting regulatory biology that controls this cycle as we go from the stolon, this underground stem, through tuberization, tuber filling, and there's a lot of sink source carbon partitioning, which centers the environment. And then the tuber goes into a period of dormancy, harvest, and then sprouting. And of course, potatoes are stored on an industrial scale, and control of this dormancy and sprouting mechanism is very important, especially with the withdrawal of sprout inhibitors. So genetic approaches where you can breed for different dormancy characteristics have come back into focus. In fact, this is where we started looking for genetic factors that control the sprouting reaction. Now, these graphs show sprout growth curves from different genotypes from the O6H1 population, and there was considerable variation. Ones with long dormancy, sprouts grew very slowly. Short dormancy ones, the sprouts grew much quicker, and we could map this trait. And we could detect QTLs on 4 different chromosomes, and these were reproducible over several seasons and of fairly large effect. And in fact, this shows a candidate gene for one of these main effect QTLs on chromosome 3, and we're particularly attracted to this gene annotated as TFL1 or terminal flower 1, and we're very interested in this gene family. It's a member of the PEBP family, the phosphatidylethylamine binding protein family. And these are signaling proteins that are very important in potato, particularly, as I hope I can show you. So we know that one of these genes encodes SB6A, which is the tuberization signal. It's regulated by SB5G. It's a negative regulator of 6A, so this is part of a complex control mechanism. Rather less is known about this TFL1 or centroradialis clade. These are known to be flowering inhibitors in other plants, and also in other plants they have a role in bud and dormancy growth control. And probably the most famous example of a TFL1 is the self-pruning gene in tomato, which is mutations in this gene give rise to a bushy phenotype in which the fruit ripens synchronously. And this mutation is present in most commercial tomato varieties. So it's a very important mutation that controls determinacy in the plant. The mother of FT clade, we don't really know what they are doing in too much detail at the moment. So we were interested in the TFL1 gene underlying the sprout growth QTL. And one of the approaches we tend to do is to make a transgenic plant where we knock it out by RNAi, or we overexpress the plant. And when we do this with the TFL1b gene, these are sprout growth curves. So the black lines are the controls and the others are various independent transgenic lines. So we knock out the gene, we accelerate sprout growth. Conversely, if we overexpress the gene, again, the controls are in black, we slow down the sprout growth. So that's the sort of phenotype we would expect if the candidate gene identified is causal for the trait. But also had interesting effects on tuber yield and development. So the overexpressing lines, the tubers develop much slower, and tuberization, the tuber yield was much lower. And in the wild, in the RNAi lines, tuberization was accelerated, and in some cases, tuber yield increased. So this is the model for how the SB6A, which I hope you can see, this is SB6A It's synthesized in the leaves, and it's regulated by various circadian clock elements in response to environmental signals, particularly day length and temperature. And the SB6A is transported via the phloem to the stolon tip, interacts with a factor we call X in this diagram, and then this induces tuberization. So the complex in the stolon tip has really recently been identified. It's the tuberogen complex, so it contains the SB6A. So the SB6A, these PEBP proteins, they're not transcription factors, but they do bind to transcription factors. So this protein called FD is a BZIP, leucine zipper transcription factor, and this complex is held together with a 14-3-3 protein. And when this complex forms, it induces tuber identity genes. And this is very similar, very analogous to the Florigen complex, where we have an FT protein binding to FD with a 14-3-3 protein, and then this induces the homeotic genes that lead to flower development. But in the Florigen complex, it's also been identified that there's action of a terminal flower 1 protein, which can compete with FT to bind FD. And when TFL1 binds to FD, the homeotic genes are repressed rather than induced. So it's a negative regulator of flowering. And we believe that the TFL1B may act in the same way. So for this model to be correct, it has to interact with FD or the FD-like 1A that's expressed in the stolon. And this yeast 2 hybrid assay shows that it does. And these amino acids highlighted in red have been shown to be the necessary ones for 14-3-3 binding. So when we mutate these amino acids, we disrupt this interaction. So it looks like this complex, this TFL1B, is a negative factor in the tuberogen complex. And this is confirmed with bifluorescence complementation. So we can see that the TFL1B and FDs are interacting in the nucleus. The mutant, it doesn't. And this is a close-up of the nucleus. And they also interact with the 14-3-3 proteins. So this is the model. The TFL1 can bind to this complex in competition with the SB6A. And there's an autocatalytic cycle that boosts the level of SB6A once this complex is formed. And we believe that the TFL1B can interrupt this amplification cycle. So in fact, if you look at the expression level of the SB6A, the key tuberization signal, it's much, much higher in the RNA-I lines than in the wild type. This is a log scale. And we thought with this increased amount of the SB6A signal, the plants may be more heat tolerant, because we know that the heat can remove the SB6A signal. And in fact, the plants do yield higher compared with the wild type at elevated temperature, but also we get higher initial yield, certainly in at lower temperature as well. So this all fits into the model. But we also thought that we could look at the transcript levels in these transgenic lines in the stolon when we've inhibited the RNA, inhibited the TFL1B by RNA-I, so that the stolons are primed to tuberize. And what we saw was a whole family of GERMIN like transcripts were significantly upregulated in the RNAi lines compared with the overexpressing lines. And this had never been reported before, so we were intrigued by it. And the question we asked, is this GERMIN actually a target of the tuberogen activation complex? And so we did transactivation experiments in collaboration with Salome Prat in Barcelona. So here we made a complex a construct consisting of the promoter of the GERMIN coupled to luciferase, which is transfected into a tobacco leaf, or Nicotiana benthamiana, and then we can add other genes to the same cells. So when we reconstruct the tuberogen complex with FD and SB6A, we see a big increase in the expression of the GERMIN. If we use the inhibitor, the TFL1B, or SEN as we call it here, we see a reduction. The mutated form of the TFL1B does not cause a reduction. And we see the same with the FD and the FD-like. In fact, higher level of expression with the FD-like, and this is the one that's expressed in the stolon. So this is fairly good evidence that the GERMIN is transcriptionally regulated by the tuber activation complex. And so we made transgenic lines with the potato StGERMIN. When we overexpress this line, we see very rapid early tuberization. And in fact, the level of early tuberization is proportional to the expression level of the GERMIN. But we also see above ground phenotypes. The plants are very heavily branched, and so you can see that the axillary buds are activated in the overexpressing lines. They're not in the controls. See in a clear difference between the wild type and the transgenic lines in this slide. And we also see an effect on tuber sprouting in these overexpressing lines. So we see these much higher number of sprouts in the GERMIN overexpressors than in the wild type, where there tends to be one main sprout. There are several sprouts in the GERMIN overexpressors, quite a significant effect. So it seems that all the axillary buds are activated when we overexpress this gene. So how is this gene acting? We don't know what the GERMIN is doing. This shows where the GERMIN is expressed in the cell. So this is the endoplasmic reticulum that's lit up when we express GERMIN RFP transiently in benthamiana cells. And these dots here are the position of the plasma desmata stained with aniline blue. So there is some evidence in the literature that some GERMIN-like proteins are targets to the plasma desmata, and of course the ER passes through the plasma desmata, so it could indicate plasma desmatal function. But stronger evidence for this came from this experiment where we bombarded GFP sporamin into tobacco cells, and we see that the sporamin is pretty much confined to the cell where it's bombarded. Less than 10% of the cells do we see these dots representing sporamin move out. However, if we co-bombard with the GERMIN, we see these spots moving out of the cell. So we think that the GERMIN changes the size exclusion limits of the plasma desmata, allowing the GFP sporamin to move to adjacent cells. And so we think that this links GFP links the GERMIN with the plasma desmatal gating with the tuber activation complex. In fact, it's been known for some time that sympastic gating is important in the control of potato tuber life cycle. So this is work from Rob Hancock at the James Hutton Institute, done with Roberto Viola and Ali Roberts. quite a long time ago now. And you can see that the plasma desmata open, allowing symplastic unloading on tuberization. The apical meristem becomes symplastically isolated. And on dormancy release, we see the converse. We see the apical tuber bud regaining connection with the flow as it starts to grow. So it's interesting that we've got this link between the tuber activation complex and these plasma desmata effects. So to summarize this part of the talk, tuberization is an irreversible commitment to vegetative growth, and therefore highly regulated. And there's many negative and positive signals that respond to the environment. I think we've shown that the TFL1B is a negative regulator of the TAC complex. Its removal accelerates all aspects of the tuber life cycle, and possibly is a good gene editing target. And the TFL1B transgenic lines enabled us to identify GERMIN as a TAC target, and the GERMIN links the plasma desmatal gating with TAC regulation. And we also think that other TFL1s are likely to have important functions, and this is just if you need further convincing that the PEBP family is very important. Looking at the sequences of the potato TFL1Bs, there's a very important part of the sequence which forms this external loop. It's called the external B loop in exon 4, and this is important for many protein-protein interactions. And if you line up the sequences of this region, we can see that there's one particular TFL called TFL1C on chromosome 6, which is identical to the tomato cell pruning gene that's so important. And the self-pruning gene in tomato controls determinacy. And so we've made transgenic lines of the TFL1C, and we want to see what effect this has on root structure and determinacy, and other related traits such as water use efficiency and nitrogen use efficiency. So I think in summary, there's considerable scope for genetic improvement of abartic stress tolerance. I think it's important to understand the tuber life cycle, because this is of key importance. I think the TFL1s are good targets for gene editing, and I think when we have the APGC facilities, will greatly expand our capacity for investigating these tuber developmental traits. So it just remains to thank and acknowledge many colleagues, with apologies to any I've missed, but Laurence and Ray have worked with me for many years and provided unstinting effort and work, as have many other of these colleagues, and former colleagues also should be acknowledged. Externally, we've worked with many collaborators across Europe and within the UK, and our African colleagues in Malawi and Kenya. And we're grateful for funding from quite a range of sources over the past decade or so, including EU project that we're running at the moment called ADAPT. So with that, I'd like to thank you for your attention, and happy to answer any questions you might have.
Thanks for that, Mark. That was a hell of a lot of information packed into that time. That was good. Is there any questions from the audience? Anyone want to put their hand up and ask something? If not, I've got a few I can ask. Am I seeing any hands going up? Hold on, give me a minute. So, okay, Mark, I can maybe ask some. Going back to the heat shock work, what's the mechanism and how that works? Do you know? Or can you speculate?
I think heat shock proteins are chaperones that basically bind to proteins and protect them from unfolding and denaturation on exposure to elevated temperature.
I mean, presumably, if you wanted to manipulate that, and I think what struck the mind in me is the loss of adaptation. It must be a resource commitment by the plant to do that. So it's just like running on full tilt all the time.
Exactly. So I mean that's why they're induced by heat.
Yeah.
I guess. But I mean I think the thing about the A2 allele is it's turned on very quickly compared with the others. So I think this can be an advantage in certain environments.
So in some of the other ones now, if you're talking about the TFL and the GERMIN thing, they're so integrated in many other aspects of the plant. How would you be able to manipulate them without changing everything else?
Yeah, that's. I mean, we're quite surprised with the TFL1Bs that there wasn't such a dramatic pleiotropic phenotypes when we knocked them out, because we were knocking them out constitutively. So it was interesting, and perhaps that makes them good candidates for gene editing. So I mean, these families have arisen from duplication and neofunctionalization, so they're quite specialized to certain tasks in the cell. So
and is there a generalism? I mean, you talked about tomato there. What about the rest of the solanum species? I think there's transferable approaches across a lot of them. I mean, if you're looking at things like peppers, for example, I mean, the areas they're grown in are getting incredibly hot as well.
yeah, I mean, I think the heat tolerance from the TFLs is collateral, but I think to have I don't know if there's any self-pruning pepper mutants that would be of interest, or it'd be interesting to see if you can make those mutations. I know it's very difficult to transform peppers. I don't, but
Yeah.
could be some interesting phenotypes.
So we've got an interesting question come in, and it's kind of going down the route I would go down as well. Could you maybe give us a bit more detail about how this will help farmers, and giving some examples of how you think this might work, and how long till we would see the utility of this science in the field?
Yeah, well, I mean the TFL1B, which is an accelerator of the tuber life cycle. When we knock it out, we can get mini tuber sized potatoes in 45 days, compared with 75 or 80. So if you're a seed producer, you could halve the time of your production if you had an edited line or used some other RNAi approach. Otherwise, some of these alleles, if I was a breeder in a warm environment, would I want the A2 allele in my potato? I think I would. So there's the breeding approaches, and then the other TFL, the TFL1C, that we think could be controlling determinacy. I think that could be critical breeding target for the future, because that's related to water use traits, nitrogen use traits. So I think that could be critical. How long it take to breed conventionally, you're looking at 10 years, but if you're doing gene editing approaches, that may be viable in the not too distant future, a lot quicker.
But presumably a classic GM approach is not one you would want to go down, because it looks like these things are so interrelated in other mechanisms and functions that you'd maybe want to knock it out for a time rather than completely out some of these things.
Yeah, having control over when you can express these would be ideal. Yeah.
So Raoul's asked, do you know if the mechanisms of tolerance to high temperatures are similar to those for cold. So can you acclimate to low temperatures for potato?
Well, frost tolerance is a big issue. I don't think it's the same pathway. I think there's a whole different raft of frost tolerance genes. It's something we've had a quick look at in the O6H1 population, actually, but we couldn't find much variation for frost tolerance.
Mm-hmm.
But again, it's quite difficult to phenotype with the, without having specialist kit.
Yeah. Okay.
Or living somewhere cold.
Well, hopefully, I think doing APGC will give you frost ability. It'll go down to cold anyway, that's for sure. Okay, so there's no more questions, and I think we've grilled Mark enough. So if you'd like to show your appreciation in the usual manner, thanks very much, Mark. That was a thoroughly enjoyable almost an hour.